Hello and welcome to G Fun Facts Online. Today, we are going to dismantle one of the most persistent metaphors in modern medicine. When you think of antibiotics, you probably imagine a nuclear option. It is the scorched earth tactic. We pop a pill, and it goes into our system like a biological carpet bomber, wiping out the bad bacteria, but often taking the good guys with it. We are told constantly that antibiotic overuse leaves our microbiome shattered and our immune system weak. We call it being immunocompromised. And while that narrative is true, it is also dangerously incomplete. Because there is a hidden side to these drugs that science is just beginning to understand. A growing body of research has uncovered what we call the antibiotic paradox. It turns out that some antimicrobial drugs do not just kill pathogens, they act as field commanders. They recruit, train, and supercharge your own immune system. They turn your cells into killing machines. They calm down inflammation storms. They are not just silent assassins working alone, they are partnering with your body. Today, over the next 20 minutes, we are going to dive deep into this paradox, looking at the specific mechanisms, from lazy lysosomes to oxidative bursts, that are reshaping the future of medicine. Let's start with a discovery that completely changed how we view the relationship between drugs and our white blood cells. This story centers on a drug called bedaquiline. Bedaquiline is a heavy hitter used to treat tuberculosis. For a long time, we thought we knew exactly how it worked. The textbook definition says that bedaquiline targets the bacteria's energy production. Specifically, it inhibits an enzyme called ATP synthase in Mycobacterium tuberculosis. Without ATP, the bacteria starve and die. Simple. Case closed, or so we thought. Researchers began noticing something strange. Bedaquiline seemed to work significantly better inside a living human body than it did in a glass petri dish. If the drug was just a chemical poison, it should work roughly the same in both places. The missing variable was the host, us. In a landmark study, scientists realized that this drug was triggering a profound change in macrophages. Now, macrophages are the large white blood cells that act as the cleanup crew of the immune system. They engulf, or eat, pathogens. But here is the problem. Sometimes, macrophages are lazy eaters. Bacteria like TB are evolved to survive inside these cells. They prevent the macrophage's stomach, which is an organelle called the lysosome, from fusing with the compartment holding the bacteria. So the bacteria just sits there, safe inside the immune cell. Enter bedaquiline. This drug acts as a stimulant for the macrophage's digestive system. It flips a genetic switch, activating a master regulator gene known as transcription factor EB or TFEB. When this switch is flipped, the macrophage wakes up. It starts generating more lysosomes. It fuses them aggressively with the compartments hiding the bacteria. The drug transforms the macrophage from a passive vessel into a hyper-efficient killing machine. It empowers the body to destroy not just TB, but potentially other bacteria that the drug itself cannot kill directly. This is the essence of host-directed therapy. The drug isn't doing the heavy lifting. It is handing the immune system a sledgehammer. But boosting aggression is only one side of the coin. Sometimes, the immune system is too aggressive. It causes collateral damage. This brings us to our second chapter, the peacekeepers. To understand this, we have to go back to a medical mystery from the 1980s involving a fatal lung disease called diffuse panbronchiolitis, or DPB. Patients with DPB were dying, despite being treated with potent antibiotics. The bacteria in their lungs, often Pseudomonas, were resistant to erythromycin, a common antibiotic. The doctors were running out of options. But then, inexplicably, they found that when they gave these patients low-dose erythromycin over a long period, the patients survived. The cure rate jumped from 20% to over 90%. This made no sense. The dose was too low to kill the bacteria, and the bacteria were resistant anyway. So what was the drug doing? It turns out, the antibiotic was not fighting the germ. It was coaching the immune system. This class of drugs, known as macrolides, which includes azithromycin, acts as an immunomodulator. In chronic infections, the body often panics. It floods the lungs with neutrophils, which are cells that release destructive enzymes. It is a scorched earth policy by our own body that destroys the lung tissue it is trying to save. Macrolides intervene here. They gently suppress this recruitment. They tell the body to cool it. But they do something even more fascinating. They help take out the trash. This process is called efferocytosis. It is the method by which macrophages eat dead and dying cells. In diseases like cystic fibrosis or COPD, dead cells pile up in the lungs. They rot and leak toxins, fueling more inflammation. Azithromycin stimulates the mannose receptor on macrophages, effectively speeding up this cleanup process. By clearing the battlefield of debris and calming the hysteria, the antibiotic allows the true immune response to function correctly. This is now standard care for cystic fibrosis, and it is a perfect example of the paradox. The drug saves the patient not by killing the invader, but by calming the host. Now, let's get a bit more chemical. We need to talk about one of the most primitive weapons our cells possess, the oxidative burst. When an immune cell wants to destroy a bacterium, It often hits it with a rapid release of reactive oxygen species, or ROS. Think of this like biological bleach or hydrogen peroxide. It dissolves the invader. We used to think antibiotics and the immune system use different weapons. But recent research suggests they are using the same ammunition. Many bactericidal antibiotics, the ones that kill directly, like penicillins and quinolones, damage bacterial structures that look like mitochondria. When this happens, the bacteria spew out toxic free radicals. But the paradox deepens. These antibiotics also induce ROS production in our own cells. This sounds bad, right? Oxidative stress is usually considered harmful. But at controlled levels, this antibiotic-induced ROS acts as a signal flare. It tells the immune system, we are under attack. It recruits immune cells to the site. It creates a synergistic kill zone where the drug and the immune cell are both firing the same chemical weapon at the target. It is a delicate balance, however. Too much ROS leads to tissue damage, which explains some side effects of antibiotics, like the tendon damage seen with fluoroquinolones. But when it works, it shows that the drug's effectiveness depends on how well it harmonizes with the body's own artillery. Speaking of fluoroquinolones, let's move to our fourth point, the recruiters. Fluoroquinolones, like ciprofloxacin, are incredibly common. We know them for their ability to unwind bacterial DNA, but they also have a surprising ability to superinduce specific immune signals. Research has shown that certain quinolones can dramatically boost the production of interleukin-2 and colony-stimulating factors. Let's break that down. Interleukin-2 is the fuel for T cells. T cells are the special forces of your adaptive immune system. By superinducing interleukin-2, the antibiotic effectively expands the army of special forces fighting the infection. On the other hand, colony-stimulating factors tell the bone marrow to produce more white blood cells. In this way, the antibiotic acts like a recruitment officer. It ensures that reinforcements are constantly arriving at the front lines. This effect is so potent that researchers have actually investigated using modified quinolones in chemotherapy patients to help reboot their immune defenses. It is a total shift in perspective. The drug is not just a poison for the bug, it is a horn blowing for the cavalry. Now you might be wondering, if these drugs boost immunity, why do we worry about them harming us? This connects to a central theme called hormesis. Hormesis is the biological phenomenon where a substance that is toxic at high doses is beneficial at low doses. This is the key to the paradox. At standard therapeutic doses, antibiotics act like nukes. They kill bacteria, but they can stun our mitochondria and disrupt our microbiome. But at sub-inhibitory doses, levels that do not kill bacteria, drugs like tetracyclines and macrolides act as hormetic stressors. They apply a tiny amount of stress to our cells. This stress triggers an adaptive response. The cells become tougher, more alert, and better at regulation. This challenges the binary view of dosing. In the future, we might see biphasic treatments. Imagine a protocol where you get a high-dose shock to kill the pathogen, followed by a low-dose tail to modulate immunity and heal the tissue. It is a fascinating concept that moves us away from simply trying to sterilize the body. And this paradox extends beyond bacteria. We are seeing similar things with antifungals and antivirals. Drugs like amphotericin B, used for fungal infections, have been shown to alter the production of inflammatory cytokines. They change the conversation between immune cells to ensure the response is robust, but not self-destructive. Similarly, the antiviral drug Arbidol, used for influenza, stimulates macrophages to eat viruses more aggressively, while also inducing interferon production, the body's natural antiviral alarm system. So where does this leave us? We are standing on the edge of a new frontier in medicine. The antibiotic paradox forces us to rethink the definition of a cure. We are moving away from the simplistic bug versus drug model. We are entering the era of the bug-drug-host triad. Understanding that antimicrobials are also immunomodulators opens the door to host-directed therapies. Instead of just designing stronger and stronger antibiotics that inevitably drive resistance, we can design drugs that specifically awaken lazy lysosomes, like bedaquiline does. We can design drugs that guide immune cells to clear debris, like erythromycin. We can design drugs that boost T-cell recruitment, like quinolones. In this brave new world, the goal is not just to kill the germ. The goal is to empower the body to save itself. The antibiotics of the future may not be measured by how well they kill in a petri dish, but by how well they lead the army of our own immune system. That is the story of the antibiotic paradox. Thank you for listening to Fun Facts Online. If you enjoyed this deep dive, make sure to check back for more content where we unravel the hidden complexities of the world around us.